Hello, my name is Grace Brubaker. I'm a senior environmental biology major at Mount Vernon Nazarene University, and this is my senior honors project titled A Study and Reintroduction of American Water Willow, or Justicia americana, in the Kokosing River. So over the course of this presentation, I'm going to go into an introduction into what American water willow is. I'm going to go over the purpose of this plant in Ohio rivers. I'm going to go over briefly over the methods that I used in this study and the results that I got. Then I'm briefly going to discuss those results, and then I will conclude this presentation. So American water willow, or its scientific name Justicia americana, is not actually related to true willows. It's called American water willow because of its superficial resemblance to true willows, but it lacks the woody stem of true willows, which are actually in the genus Salix. It's native to the eastern United States, from Texas all the way up to Ontario, and it grows in dense colonies along rivers, streams, lakes, and ponds. It has small orchid-like purple flowers. You can see a picture of one on the right with the Tanuka moth, and it reproduces both sexually through seeds and asexually through fragments, so pieces of the stem or roots breaking off, or creeping rhizomes, which is partly what allows it to make dense colonies along Ohio rivers. Over the course of this study, I sought to answer the question, is it possible to establish healthy colonies of American water willow, or Justicia americana, in the Kokosing River despite the plant's current absence? I hypothesized that because American water willow is present and prevalent both in the nearby Mohican River and downstream in the Walhanding River, that attempts to establish Justicia americana in the Kokosing River would be successful. So these plants play major ecological roles in the health of Ohio rivers. They help to mitigate erosion. They decrease the suspended sediment in the water column, which also increases the water clarity. They provide habitat for fish fry and also for the insects that fish fry eat. They provide food as well for beavers, deer, and muskrat, along with other animals. They're abundant in the Mohican and Walhanding rivers, but they're much less common in the Kokosing River, which they're kind of prevalent in some of the tributaries that lead into the Kokosing River, but they're much less common through the stretch of the Kokosing that runs through Mount Vernon. So the first phase of my study began in the Mohican River. On October 7th of 2018, I did a survey of the Mohican River from the Greer Landing access point in Glenmont, Ohio, to the Bridge of Dreams in Brinkhaven, Ohio. I surveyed 13 colonies along that, and I surveyed them for colony size, colony density, which is just how closely the plants grow together, water depth, substrate type, which is the type of soil that they're growing in, so rocky substrate or sandy substrate. The location in the river channel is just where it's located in the river, and then the level of submergence. And then I use these results to visually assess the Kokosing River planting sites to decide what habitat was best for them. And that began phase 2 of my study in the Kokosing River. I had 3 plantings over the summer of 2019. They began in March of 2019, and then I had one in June, and then August of 2019. The plants that I had were removed from the Mohican and Walhanding rivers. The first 2 were removed from the Mohican, and then the August planting was removed from the Walhanding River. I planted between MVNU and Mount Vernon, Ohio, and then Big Run Road in Gambier, Ohio. I had 15 planting sites along that area. And then I planted them based on a visual assessment of substrate type, sun exposure, and then the location in the river channel. They were shallowly buried, less than 4 inches deep, and spaced 6 to 18 inches apart, with the previous year's, that year's growth above the ground. As you can see on the right, that was my 14th colony, and that is what most of the colonies looked like after planting. I planted along the edge of islands, midbars, and banks. to allow for growth into the river as well as up onto the bank farther. In total, in the summer of 2019, I planted 416 plants, 110 during the first one, 180 during the second, and then in the August planting, I planted 126 plants. As of October of 2019, I had 152 surviving plants, which took my spring and summer survival rate to about 36.54%, which was surviving plants in October compared to the total plants. And then I checked them again in June of 2020 and had 86 surviving plants, which gave me a first-year harsh season survival rate, which is just the freezing temperatures of winter as well as the high water events of spring. And that gave me a harsh season survival rate of 56.58%, which is the June survivors compared to the October survivors. That gave me a total survival rate of about 20.67%, which was June survivors compared to the total planted. And I had 10 colonies that had less than 5 survivors. I would have considered them not successful. And then 5 colonies that had greater than 5 survivors, which I consider successful. My 2 most successful colonies are Colony 7 and Colony 13. And as you can see here, they had similar survival rates. Their health was comparable, but the main point here is that the planting conditions were very different for each of them. Colony 7 was planted on a midbar. Colony 13 was planted on an island. We have a shallow slope and a steep slope to the bank that they're planted on. Rocky substrate versus sandy substrate. They had a similar amount of sun, but Colony 7 was unvegetated, while Colony 13 had black willow previously growing on it. And here are pictures of those colonies. As you can see, Colony 7, it has some bigger rocks and stuff that it's growing in between. It's not very dense, but you can see on the left, just underneath the surface of the water, there are some underwater rhizomes that are spreading. This means that it's healthy and reproducing. And then on the right, you can see the emergent growth, and it's starting to form clumps that you can see, which is what it's supposed to do. And this seems to be its growth form in the rockier substrates. And here you can see Colony 13, which visually was the healthiest colony. It had flowers in June of 2020, and it's starting to get denser and bushier than a lot of the other colonies. But on the left, you can see the creeping rhizomes, which are very healthy, and they're very apparent in that picture. And then on the right, you can see how the growth is getting denser as it gets older and grows. So my hypothesis was supported. I had several colonies that were successful. They're doing very well, flowering and reproducing. But my colony survival and growth was noticeably impacted, mostly by its resistance to erosion, the site's resistance to erosion. Established vegetation plays a role in a site's resistance, as well as large substrate, like you saw in Colony 7. The larger rocks didn't move around as much, which allowed the colony to take root easier. Steep banks, like in Colony 13, keep the plant elevated out of the water a little farther, so in high water events it's less likely to wash away. And then sheltered areas were mostly evident in the Mohican survey. I was able to see lots of colonies that were in sheltered areas away from the currents and places like that, but that was also an apparent feature of some of my other successful colonies. And then sun exposure also played a part. I think 4 of the 5 colonies that were the most successful were in part to full sun. There was only 1 that was in shade. To expand the idea of resistance to erosion a little, because this appeared to be the most important factor in colony survival, all colonies with a 0% survival rate in October of 2019 had sandy or silty substrate. And as you can see there on the right, Colony 3 was one of the siltiest areas I planted, and they were very healthy October of 2019, but by June of 2020, most of the plants had washed away, and you can see very obviously near the top of that picture the line where the river eroded away that bank. June 2020 colonies, colonies that had plants in June. The colonies that had rock or gravel substrate had much higher survival rates at 74%, 90%, and 140%, which just means that the plant reproduced, versus colonies that were planted in sand or silt, which had lower survival rates of 28%, 43%, or 50%. So other factors that could have played a role in the health and survival of American water willow in the Kokosing, both previously and during this study. Invasive plants. Japanese knotweed and bush honeysuckle are very prevalent along the Kokosing River, and they can offer a lot of competition for sunlight along banks. Not as much on sandbars unless it's a pretty vegetated sandbar already. But those are also present in the Mohican and the Walhanding rivers, so that didn't offer any conclusive evidence. Previous acidic conditions. American water willow can't survive at a pH below 5.5. So if the river had previously dropped below that, it could have killed historical colonies that were already there. There are several unlikely factors that have been known to affect American water willow populations, but we don't see evidence for these in the Kokosing River, including pollution. The Kokosing River is a state scenic river, and it has populations of eastern hellbenders and spotted darters present, which are both very pollution-sensitive, state-endangered species. You can see on the right there's an eastern hellbender salamander that's just a young one that was actually being released into the Kokosing River in 2018 because it's been deemed a healthy enough habitat for these endangered species. And then foot traffic and trampling is another option, but it's unlikely because the foot traffic in the Kokosing is significantly lower than that in the Mohican. There were also several limitations to this study. There was a lack of conclusive results from the Mohican survey, and this resulted in me planting in regions of deposition because I was trying to find sheltered areas. And a region of deposition is just the region on the downside or the downstream side of a sandbar where the sediment drops out of the water. And this resulted in some of my plants being buried beyond what they could sprout from. The March 24th planting was also a little bit of a limitation because I dug up the plants while they were dormant. As you can see on the right, there's a dormant plant, intending to reduce the amount of transplant stress. However, this was very ineffective because 10 days after I planted, the highest water event of the spring came and washed all but 2 of them away. And then this study was primarily observational, which can be a little bit of a limitation. And then identifying individual plants was sometimes a little bit of a struggle because these plants have a clumping habit, and so sometimes it's difficult to tell what is one plant and what is just several stems that have grown close together. Overall, however, this reintroduction was a success. Erosion appears to be the biggest threat to small colonies, but it should not significantly affect established colonies, and current conditions don't prevent the growth and survival of Justicia americana in the Kokosing River. Lastly, I'd like to give a special thanks to Brian Zimmerman, who introduced me to this plant and gave me this idea for this study. Catherine Suggs, who's pictured there on the right with an Ohio lamprey on her arm. She accompanied me on many of my kayaking surveys. Thanks to Hannah and Laura Brubaker, my sisters, for also accompanying me. And then thanks to Jonathan Brubaker, my brother, for editing and putting this video together for me. And lastly, thanks Dr. John Bosley, Dr. Dorothea Hawthorne, and Professor Chris Ingersoll for taking the time to read and edit and give feedback on my paper. I really appreciate it. These are my references. Thank you all so much for listening to my presentation. I hope you all enjoyed it. If you have any questions, please feel free to leave them in the comments. I will do my best to answer them. Thanks.